and I'd like to introduce my student, Georgia Chamblee. She worked on this project primarily last semester. We're continuing the project this semester, but she also took my modeling class last semester, and so she's really interested in this particular receptor, and I'm going to let her explain why to you. Okay?
I decided to do the L-dopa in reacting in the aromatic L-amino acid decarboxylase enzyme. I became interested in it because of its prevalence in a lot of neurological conditions, one of which I was diagnosed with last year. So for this project, we downloaded the crystal structure of the aromatic L-amino acid decarboxylase enzyme, and we placed that into Mo. It had a lot of structural issues, missing atoms, and charges were incorrect. So we fixed those using the structural preparation sequence in Mo. We couldn't find a specific bonding pocket in the enzyme, which contains 2 chains. So we selected the entire pocket inside the enzyme and docked the L-dopa in that. Here's a picture of the completed one. Originally we had 6 complexes, and from that 5 of them were in the same area, which allowed us to locate what we believe to be the specific binding pocket. Here it shows the interactions in the enzyme. We ran dynamics on it, and we got the most, this was the chart with the most interactions that showed the strongest interactions in the enzyme. So from that, we were able to gain some unique information on what interactions happen, which amino acids interacted from the enzyme with the L-dopa, and we haven't really been able to find any computational models that have been published showing this so far. We were able to find that the aspartic acid, we found particular links in the interactions with the groups in the L-dopa. Here we had aspartic acid A310. It interacted at a distance of 1.79 angstroms. We had lysine A361, and that is here, interacting with this oxygen at a length of 1.82 angstroms. We had arginine interacting at 1.68 angstroms, arginine A356 interacting at 1.92 angstroms. We had aspartic acid again, A92, interacting at 2.09 angstroms. We had an aspartic acid from the A chain that reacted at 2.29 angstroms, and an aspartic B92 interacting at 1.86 angstroms. So we were just doing the computational model. We haven't found mechanisms so far. We're just working on docking studies at this point. This was our first, we're planning on continuing some more docking studies to find more specific information.